And I'm very excited for the next session. We have three great speakers. And the first speaker is Dr. J.T. Elder, who's the Kirk D. Wuepper Professor of Molecular Genetic Dermatology and the Director of the Training Program in Cell and Molecular Dermatology. And he's going to present the use of MGI to study TRAF3-IP2, a gene strongly associated with psoriasis, and its role in pro-inflammatory T helper cell polarization.
Thank you very much. So psoriasis is a very common disease. It affects 1% to 2% of the population. It has associated nail disease and painful arthritis that develops in about a quarter of patients. Psoriasis has actually played a major role in the development of genetics over the years, going all the way back to the studies on the Faroe Islands by Gunnar Lomholt. It's also one of the first diseases to manifest HLA associations. So we've been working on this since the early 1990s, initially with families, and more recently with cases and controls. And this is a result from a couple years old now that shows you our plot, our Manhattan plot. And we are just about, by working with many other collaborators, just about to be done with an expanded GWAS that's going to take us up to a total of about 140,000 participants. And that should take us up to where we have like 180, 200 genes. But also important, it will narrow down the 95% Bayesian credible interval for regulatory signals, because that's, like everybody else, that's what we've run into. We're also lucky to have a new data set from Regeneron on we have almost 10,000 samples, and so they've all been GWAS'd and exome sequenced. But this story is familiar to all of us. You know, most genetic signals seem to be regulatory in nature, and so you can't necessarily assume that well, first of all, there's linkage to equilibrium, but then there's also the problem is you don't know if the genetic signal is right next to the gene that you are wanting to find out about. We do know this is some work from Alex Soy, who was in the department, still is, where basically he was doing gene ontology type analyses and also H3K27AC chromatin, active chromatin studies. And the story comes out, pretty evident that even though we don't know exactly what the genes are, we know that the immune system is involved. So I have two little stories. I have way too many slides. I apologize. I want to show you what we're doing in general to try to understand our signals has to do with basically calling people back. And we usually work with blood, and then we've developed some cellular systems that we can get using people's blood. And we've used it to study some regulatory genes and also this TRAF3-IP2 structural gene. So many of you are, I'm sure, familiar with ATAC-seq. We've been doing it for about 4 years now. We've gotten pretty good at it. And what we're basically getting is a map of open chromatin across the GWAS. We are then coupling that across the genome. We're then coupling that with a GWAS genotyping on everybody. We're hoping that we can have a decent power with about 150 people. About half are normal and half are psoriatic. What we've really done for the most part is we've been able to rely on our own collection. But as I'll show you in some of the cases, we have some rare alleles that we'd like to study. And even our own collection of 9,600 people doesn't have enough examples of rare heterozygotes. So we're about ready to go with this. We've got our data freeze, and we're going to be, and we're doing a lot of sequencing right now to get chromatin QTLs and expression QTLs. So this is one of our systems. Basically, we start with PBMCs. We separate out myeloid and T cells. We then use flow cytometry to separate different subpopulations of T cells. And we do a day of CD3, CD28 activation to basically activate T cells. And then we either have RNA-seq or ATAC-seq, and of course the GWAS genotyping. And this is just an example of how we can get our cell populations on the flow sorter. Now Alex came up, this is just sort of a shortcut. When we have our GWAS chips back, we will basically know, you know, what all the variants are for all our people. But in the meantime, Alex has been using just the sequence data that has come out from the open chromatin data to look for people who are heterozygous for variants that might affect open chromatin, and that has worked actually very well. And I'm not really going to be showing you any of the data from MGI today because we're still doing it, but we are excited because we've been able to find quite a few genes that have differential chromatin accessibility as demonstrated by ATAC-seq, and are also mapped very close to GWAS hits. Here's an example for activated T cell, CD8 positive activated T cells, that 10 out of the 11 variants that we found in the activated T cells that map to psoriasis 95% confidence intervals are also blood cis-EQTL. So we got that information from the EQTL Gen Consortium, which is a really nice resource. So here's one example, and this is the kind of idea here that when we have an area of interest like this, we can go ahead and use MGI and pick people out who have the genotypes that we want. So this is called the IL-28RA is the old name, now it's interferon-like receptor 1. And you can see that there's an area where we had accessibility peak show up, and the most strongly associated SNP in the region is right on that peak. And that SNP is also an EQTL for the gene. So we're excited that, you know, even though this is a global approach, that we're coming up with hits that we can then follow up in other data sets for people or other people that we can actually call back. Of course, the issue here is, you know, many of these allele frequencies are not great, and so especially if you're trying to look at all three genotypes, the people who have the, or homozygous for the rare genotype can be very hard to come up with. But you know, with the 70,000 people that are all genotyped or will soon be, we can easily find those people. And we have worked with MGI to set up a system whereby we have a list of all the genotypes. We send them to MGI. MGI finds the people and helps us contact the people. And then we enroll them in our study, our own psoriasis genetic study. And that's, it's working. You know, I don't have exact numbers for you on how often people are coming back, but the mechanism is in place. So this is the biology of this gene. It's quite interesting. Another gene that's really interesting is, that it makes another point I want to make, is the IL-23 receptor. So if psoriasis is very effectively treated by drugs that target the IL-23 and also drugs that target IL-17 or its receptor. And one of these, and it's also kind of stacked up with a bunch of different psoriasis risk, known psoriasis risk loci, like JAK2, STAT2, STAT3, and STAT5 are downstream members of this pathway. And actually, we have also worked on psoriatic arthritis, and we do have two different genetic signals in this locus, one for cutaneous psoriasis, or PSC, and the other one for psoriatic arthritis, or PSA. So this just shows you on a locus zoom plot where the two peaks are, and the arthritis signal has also been seen in ankylosing spondylitis. The cutaneous psoriasis signal actually seems to be an expressed variant called R381Q. So now I'm just going to zoom in on this a little bit more. I'm very excited about this data. So the cutaneous psoriasis association is on the top line. Then the arthritis association is on the next line down. So you can see it's more 5' on the gene. And what I'm really excited about is the cis-EQTLs from the EQTL Gen database just map exactly to, they look just like the association p-values for psoriatic arthritis. And these are H3K27AC maps down here, and you can see, as we know, there's a lot of gut diseases that are associated with arthritis as well, and psoriatic arthritis. And this gene seems to have one regulatory region that is active in colonic epithelium, another one that's active in mesenchymal stem cells, and yet a third one that seems to be activated in T cells. That's this one here. So this is just showing you that we have activated related open chromatin in our T cell subtypes in this region as well. And I think one of the reasons it's been hard to sometimes really put your finger on exactly what a GWAS signal means is because you could have different cell types that are all part of the disease process that could have different regulatory regions in the same gene. Okay, so I'll finish up. Yeah, I'm doing okay. I'm going to finish up talking about a gene called TRAF3IP2. It's interesting to us because it encodes. It's downstream of the IL-17 receptor and couples interleukin-17 to downstream cellular responses. And there is a coding variant that seems to make sense here. Position 10, that is part of the interaction domain with TRAF6. And so what's curious about this is the allele that's supposed to be risk is actually, for this particular molecular phenotype, is actually loss of function. So that's weird, and there's a lot of stuff we don't understand about that yet. But I'll just, I want to bring this up in the context of some cell biology because, again, this goes back to what we can use MGI resources for. We can call people in, we can get blood. It's not that hard to do. We'd like to do skin, but skin has some technical issues. But anyway, neutrophils are the shock troops or the infantry of our immune system. They only live for a day, and people basically don't really like to study them because all they ever do is show up and blow up. They're really hard to work with. And so that I always call these cells the Rodney Dangerfield of the immune system. Doesn't get enough respect. So you can actually show that there's stuff in psoriatic serum that will activate these neutrophils to make what's called neutrophil extracellular traps. You can actually take normal neutrophils and take the serum from a psoriatic person, and depending on the severity of their psoriasis, they will increase the amount of nets that can be formed in vitro. And so this was Sylviane Lambert's work and Kaylee Hambro. Basically, again, starting with red blood cells, you have a 7-day assay. Now, not just 24 hours, but now 7 days. And what we're doing is we're taking the neutrophils out, putting them on the dish, and allowing them to form nets, and then we're just dumping the rest of the the peripheral blood mononuclear cells on top of them. I wouldn't be talking to you about it if it hadn't worked. So this is a demonstration that the percentage of what are called Th17 cells, which are CD4 positive T cells that express IL-17, is increased by nets and further increased when we show a tiny bit of LPS to the nets. You don't really have to don't have to use LPS, it just kind of helps a little bit. And another interesting piece of biology here is this is completely needed. Presence of myeloid cells is completely required for this because if we take out the monocytes when we purify the cells, nothing happens. There's two kinds of netosis, which I'm going to show you, but we were asked to make nets in more than one way by our reviewers, so we finally did. And what happens is that both PMA and LPS will make more neutrophils and turn into nets. And in fact, PMA is actually better at this than LPS. And yet, when we make our nets with PMA instead of LPS, we completely lose the ability to polarize Th17 cells. So as far as how that relates to nets, it's now thought that there's two modes of netosis, one called late suicidal and one called early vital. And in the early vital, the stuff that's inside the neutrophils seems to get packaged up into membrane-encoded vesicles and released from the cells without killing or breaking open the cells. So we're starting to think about how we can get at that difference between PMA nets and LPS nets, maybe in terms of looking at the effect of vesicles. So where does genetics have to do with all of this? And this is where our first project that's using the MGI comes in. Phil Stewart, who's here, helped me do this analysis. So these are those same 4 conditions, except now we have 3 different kinds of people. People who are homozygous for the ACT1D10N mutation, people who are heterozygous, and people who are nullizygous. And we got a significant difference between the groups. And so this was only 6 people in the homozygous group, and that's where we're using MGI resources to make this group bigger and make sure we're not missing something. So in conclusion, we are up to, in the GWAS of psoriasis, it's up on the order of 100,000 or more individuals. We have over 80 loci with more to come. Psoriasis genome is an immunome. Most of the signals are regulatory, and what we're trying to do is understand the nature of those variants. And by just sort of using the effect of nets as a biological fire alarm, you might say, we were able to demonstrate a genetic effect of this ACT1 gene on Th17 expansion or polarization that's genetically important. So I'd just like to thank all the contributors from many different places. And Phil Stewart's in the back if you have questions. And that's it.
What are you going to study in the patients that come back? And can you make, like, for example, iPS cells from them and figure out if they respond to different meds, or if there's, to what extent are you phenotyping the people that are coming back from MGI?
I have not actually thought about making iPS cells. We certainly could. If we could get a skin biopsy, we could start with fibroblasts, but that's kind of a lot of work. I basically, if you saw the 2 assays that I described to you, we're bringing them back in and running those assays on them right now. That's what we're doing.
Do you have any kind of estimate about your success rate of getting people back?
I can't tell you. We've got the lists, and we're in the process of doing it, so I don't have that information. I'll certainly let you know when we do.
It'd be good to have.
Thank you.
Thank you very much.